So I've talked before on here about cryptobiosis in certain animal species like rotifers and tardigrades, etc. But I haven't really talked kind of about mechanism. And it turns out, I just learned this recently, that there's actually a few different mechanisms in like how they do it, depending on what type of animal it's done. So specifically anhydrobiosis, which is surviving very low water levels. So it turns out the anhydrobiosis response is actually a result of convergent evolution because it's done by a bunch of different mechanisms in the animals that do this anhydrobiosis. So one of the first mechanisms for anhydrobiosis was through the use of this molecule, trehalose. Or trehalose, I don't know, however you want to pronounce it, but trehalose. So it is a type of carbohydrate, so basically it's just a sugar molecule. So the genes for producing trehalose were found in specifically nematodes and brine shrimp like this. And so what happens when these guys undergo anhydrobiosis, they dry out, and the trehalose does this process called vitrification. And vitrification is described as essentially turning into glass. So it's not actually glass like in the windows and stuff is made of silica dioxide, and this is not made of silica dioxide, it's made of that sugar, trehalose. So it's a glass-like substance made from the sugar, formed via the process of vitrification, but for all intents and purposes, it's described as turning into glass. So that is a very neat process, and for a while it was thought that all creatures that do anhydrobiosis do a similar process. Another group of animals that does anhydrobiosis is the rotifers, and they do not have the genes for producing trehalose, so they produce late embryogenesis abundant proteins, or LEA proteins. And these, I didn't look too much into how they actually work, but they do a similar thing of preserving the rotifers in order to survive desiccation conditions. And in the case of tardigrades, they use their very own specific proteins that have so far only been found in tardigrades. And these are the tardigrade disordered proteins, or TDPs. And there's actually a group of 3 different proteins that form the complex that makes up the TDPs. So how these work, normally when the tardigrade is in hydrated conditions, so plenty of water, they're just hanging out, they are disordered, so the tardigrade disordered proteins, and they don't have any specific orientation, they're just kind of floating around, well, not floating around, but they're disordered, they're chaotic. However, when the desiccation process begins and there is less water available, the tardigrade begins to process into a ton, which is the dehydrated version, and these proteins condense and form this kind of like capsule barrier shell thing, which works to protect the tardigrade. And most of the sources I have seen have described this as the process of vitrification, so turning into the glassy substance without it actually being glass, but instead of it being due to the saccharide, so the trehalose, like in the, what were they? The brine shrimp, that's the bitch. So instead of using the trehalose, like the brine shrimp, they use these special TDPs. However, I did read one source, a more recent one, that said it may not be vitrification, but that was the only source I could find, and a bunch of other ones said it is vitrification, so take it with a grain of salt. So yeah, they use these TDPs in order to form this complex around them that protects them from desiccation. So it's very interesting that this process has come about multiple times through multiple different mechanisms. and like in the water bears, in the rotifers, in the brine shrimp, the nematodes, all of these guys, and they all tend to get lumped together as look at these crazy things, they can survive these extreme conditions when it's happened multiple times and like arose through a bunch of different mechanisms. So I don't know, I thought that was cool.